emphasise that this work has been done in 2014 and 2015, and it's a collaboration between Clarity Pharmaceuticals, the Peter MacCallum Cancer Centre and Sir Charles Gardner in Perth. Okay, so this is a summary of what we're going to be talking about today. A lot of the slides have actually been sort of introduced over time, so I'm going to get straight into it. Thanks very much, Professor Rod Hicks, for introducing this slide at the pre-meeting and today. This is a comparison of Optimised with copper-64 dotatate. It was done out of Copenhagen with Andreas Kerr's group. What we're seeing here is that copper-64 dotatate shows greater imaging clarity over the indium-111 agent. This group then went on to do a further trial, and they did a head-to-head comparison of the two imaging agents, and it showed in 75% of patients more lesions were detected, and this was with imaging at 1 hour. Obviously, changing patient imaging also changes patient outcomes. However, what they also reported was that the use of DOTA as the copper-64 chelator led to poor late retention of copper-64 in areas where there were known to be tumour deposits. So you can see in this imaging that at 1 hour, 3 hours and 24 hours, the uptake of copper-64 is actually lessening, and there is an associated increase in copper-64 detected in liver. Now this is fine for this group because they do all of their imaging at 1 hour, but if you're thinking of performing prospective dosimetry results or looking at copper therapy using this molecule, it's actually not suitable. So this has led on to the work that Associate Professor Paul Donnelly is going to present on Wednesday morning, and he will give a fantastic talk, I'm sure, about what he aimed to do, which was to develop a superior copper chelator and then to conjugate that chelator to octreotate for imaging purposes. So what he would have been looking at was something that radio-labelled quickly, requiring no heat, being very specific for copper, and looking at something that radio-labels over a wide pH to make it quite easy to work with in the lab. And ultimately coming up with a chelator that is suitable for copper-64, copper-67 imaging and therapy pairing. So some of the work that he'll end up presenting is shown in this reference, but also we've got DOTA on the left-hand side for you, and then MecoSAR on the right-hand side. At the bottom, we can show that this is copper-64 or 67 SARTATE. That's the chelator combined with the peptide. And in those imaging, this is from a preclinical study that Paul will spend more time talking about, but comparing copper-64 SARTATE to copper-64 dotatate in mice, and it is showing higher uptake at 2 hours and 24 hours in the SARTATE molecule. So that's the background. Where I come into the work is to actually look at the development of copper-64 SARTATE for use in humans. I particularly want to focus on the radiochemistry. So the preclinical vehicle that I was given to work with had SARTATE dissolved in phosphate buffer with copper-64 chloride added. They were mixed and incubated at room temperature for 20 to 30 minutes. At the end of that time, radio-labeling efficiency was about 98%, and at later time points, they actually increased the radio-labeling efficiency to greater than 99% using a C18 set-pack solid phase extraction with elution by ethanol. The vehicle was definitely something that just gets mixed in Effendorf, so not something that we do look at and think of as being suitable for humans. What we looked at was creating a robust but manual production method. So the elements that I looked at for refining were the phosphate buffer used. I actually found that the way that I did the reaction that phosphate buffer caused copper phosphate to form, which is insoluble and also reduced the radio-labelling yield of the product. So I replaced the buffer with ammonium acetate buffer. Again, the same copper chloride was used as supplied by Sir Charles Gardner Hospital. They were mixed and incubated at room temperature for 30 minutes again, and we had purification through a C18 set pack for two purposes: to desalt the reaction mixture and remove any unreacted copper-64 cations. Again, the product was eluted in ethanol, and we used 0.9% saline to dilute this to an appropriate level. So the final product was prepared aseptically. It went into an aseptic vial and it had 0.22 micron filtration, very similar to what we see with gallium-68 dotatate production. What was important to note was radiolysis. So radiolysis has been noted in many other radio-labelled octreotate formulations using a variety of isotopes. We've already seen a bit of that today with what Oliver has presented. This paper is from the Erasmus group, and it's looking at dissociation that occurs due to radiolysis, and it's also looking at individual radiolysis quenching agents. So you can see in number 1 up there, it's a HPLC chromatogram. You're seeing some mess at the baseline, essentially, and this group has investigated with a number of different pharmaceutical productions how to reduce that. What they've generally come up with is that you need a combination of quenching agents to fully remove any radiolysis. So with reference to this work, we definitely saw radiolysis in copper-64 SARTATE, and we wanted to limit that, seeing as it was an investigational drug. Firstly, it can only be properly assessed by HPLC. TLC is great, but it's really only good for assessing the percent of activity bound versus percent of activity that is free copper-64 cations. You do need to have slow isocratic elution on your HPLC. If it's too fast or if it's a gradient, you do run the risk of not actually detecting those radiolysis compounds. They can all end up in the main peak. And the number one thing that you really need is a sensitive gamma detector. Unfortunately, I don't have a blow-up of the baseline of this trace, but you can definitely see that there's a lot of little peaks. They're very small in comparison to the main peak. And again, the table shows that. And also I just want to point out that the amount of free copper-64 is quite small. It definitely is bound copper that is causing the problem. So we needed to work on refining the additional excipients added to the formulation. All of the things that we did included increasing the volume of the reaction mixture to 5 mils. Less concentration equals less radiolysis. We added ethanol to the reaction mixture. We found that a range of 4 to 10% will reduce radiolysis without affecting retention of copper-64 SARTATE on the set pack. And then we've also looked at the addition of gentisic acid along with ethanol, and this seems to be a good combination to reduce radiolysis. One thing I'd like to point out is that ascorbic acid is a great quenching agent, but it's terrible to use with copper 2+ because it reduces it to copper 1+. So I just want to show you, this was an experiment done on the same day with 2 productions of copper-64 SARTATE. You'll see that one of them has ethanol only in the reaction mixture, and the other one has ethanol and gentisic acid in the reaction mixture. Although we're getting very small peaks present, there are some peaks that do not form by having gentisic acid in the reaction mixture. Okay, so once this process was brought to a stage where we were happy with the final formulation, a site validation was performed at the Peter MacCallum Cancer Centre by Peter Ozelt and Wayne Noonan, with some external sterility testing done by Cyclotech Proprietary Limited. All of the production and quality control documentation was produced at the Peter MacCallum Cancer Centre, and 4 validation productions were completed. All of them passed completely on all tests, and that meant that we had a validated protocol that was then used in the human trial. So these results have been sort of shown, so I'll talk about them quite quickly, but 10 patients have been imaged with copper-64 SARTATE with NETs. The SARTATE was shown to be safe and it's high-level targeting. What you can see in this imaging is that there's a pancreatic tumour that shows good, quick uptake at 1 hour that's persistent up to 20 hours. You can also see that the liver background reduces over time, and that's particularly evident in the sort of grayscale imaging. This is a comparison of copper-64 SARTATE to GATATE. So we've got 1 hour imaging of GATATE and copper-64 SARTATE in the same patient. They're both showing equivalence. Copper-64 SARTATE is showing superiority at a later time point because of that liver clearance and the fact that it's still being highly retained by the tumour. And just for the last image, this person was imaged at 24 hours, and a lesion was actually seen that wasn't seen at any other earlier time points, which was very good. It was in the tail of the pancreas, and it was definitely added additional information to that person's treatment options. So just to talk about our future work, that process was a manual method. We've been working on automating the manufacture of the copper-64 SARTATE on the iPhase Technologies multi-SYN radiosynthesizer. So I'm presenting a poster on Tuesday morning about that. We've also been able to source copper-67 SARTATE from Idaho in the US, and so that has also been transferred onto the multi-SYN. What we're looking at doing is having centralised distribution of imaging and therapy radiopharmaceuticals available in 2017 for human clinical trials. And as I've said, this is all work funded by Clarity Pharmaceuticals. So Clarity is moving into phase 1 and 2A clinical trials in the following areas. So continuing with imaging and therapy in NETs in adults, but also looking at neuroblastoma imaging and therapy in children. And I'd just like to acknowledge the University of Melbourne, the Peter MacCallum Cancer Centre, and Sir Charles Gardner Hospital, who contributed to this project. And this is just a shout out to all of our collaborators as well. Thank you very much.
Thank you very much indeed, Charmaine. Any questions? There's 2 microphones over here. As people are thinking their questions, I have a quick one. Copper-64, it's got the beta minus as well as the beta plus. And those images, there seem to be some subtle pituitary uptake and even salivary gland uptake. What's the dosimetry based on this compared with the gallium? I mean, is the dosimetry favourable for these non-target tissues?
I'm not a doctor in that regard. I do think it's favourable, but I can't really present any data on that. So.
And in terms of mass that you inject compared with the gallium agents, is the mass the same?
It's a maximum of 20 micrograms for imaging,
yeah.
So it's in the same ballpark.
Okay.
Yeah.
Thank you.
I don't know if, does Michael guess, is the dosimetry favourable for?
Well, no, probably not the right phrase. I don't think there's any favourable dosimetry, but
yeah.
Is it acceptable?
We've measured the dosimetry in all the organs as part of the phase 1 trial, and hopefully be published in that very soon. But we use a lower administered activity than we do typically for a gallium octreotate scan because of the longer half-life of copper. Yep. And so those images were typically with 200 megabecquerels of copper sartate. And with
I'm just doing my 37 times table. So that's 3-4 minutes. Okay.
And it's so the but the dosimetry is very favourable compared to routinely used PET radiopharmaceuticals.
That's really good news. Any other questions from somebody else other than on the front here? No? Okay. If not, thank you very much indeed, Charmaine.
Thank you.